Hi. So I think we are live right now. Yeah? Can you hear me all in the chat? Can you say hi if you can hear me? If there is any noise problem in the... Yeah? There is some delay, so actually there will be a couple of seconds until they reply. I'm looking at the chat to see if people are... Yes. So there is a first yes. Okay. Yeah. Yeah, thank you everyone for being there. We are very happy to be here today. We have, as last session, we have Luis Puelles and Paul Cisek, and we have today with us Maria Tosches. So the plan today is to do a short summary of the first session, the first chapter of the book, and then I will present the second chapter of the book. And then we will have a discussion that will be a bit different from the previous session. I believe Alex will explain a bit how it will work. So I will let Alex now speak. So whenever you want, I believe you have to share your screen, right? So have we lost? You're muted. You're muted, Alex. I think we've lost Paul. In the meantime, you can try to share your... Yeah, mute the other computer. If you have another computer connected, mute. And yeah, Paul is here, but he switched off his camera, I believe. You're muted. Yeah. So,
Manuel, could you go out instead of Raquel? Because I cannot share the screen and she cannot go out.
Because we are too many? Okay, then I'll leave. Yeah.
Thanks. Okay, so hello everyone. As you know, we are connecting from Alicante, Spain. It is 18 in the evening, and well, just as Manuel, I want to welcome everyone to the second session of the reading club, the book Brains Through Time by Georg Striedter and Northcutt. Thank you all very much for coming, and as you can see, today we have an amazing panel of experts. We are super excited to continue this travel through this beautiful story of how the brain evolved. So I'm just going to do a quick introduction to the event, which is organized by Manuel, a postdoc working on computational neuroscience, Raquel working on systems neuroscience, and finally me studying brain development and evolution. So as you can see, we come from quite different fields, but we joined in the organization of this reading club, aiming to learn about the evolutionary history of the vertebrate brains, and especially to promote the discussion between these distant fields in neuroscience about the consequence that this history has to our understanding of modern brains. To do so, this book is ideal as it covers very nicely this natural history from early vertebrates up to primates. And we started this journey 2 months ago with the session on the first chapter as an introduction to the methodologies and terminologies that are going to be used in the book and that are necessary to follow it properly. We had a very interesting discussion with the audience. In case you missed it or you want to review it, you can find it on YouTube or webpage or in our social media. For today's session, we are going to focus on the origin of vertebrates, reviewing the main traits of the first vertebrate brains together with their most close relatives, and also the conditions under which they arise. To do this, we come with the help of this amazing panel of experts with Maria Tosches, professor at Columbia University and an expert on evolution of neuronal cell types and circuits. Luis Puelles, professor at the Universidad de Murcia and main author of the prosomeric model, and Paul Cisek, professor at Montreal University with a big trajectory in phylogenetic refinement and behavior. So, as Manuel said, in today's session, we are going to introduce some changes in the structure of the event. We will start with a short chapter summary by Manuel Molano, followed by these 3 discussion blocks. In each one of these blocks, our panelists will highlight some important or controversial aspects of the chapter and give us more insight on them in order to arise discussion with you. Each of these blocks will consist in a 5-minute presentation, followed by discussion with you. Maria is going to tell us about the origin of neurons and nervous systems. Luis is going to talk about the amphioxus forebrain, its subdivisions and its implication on function. And Paul is going to talk about the lamprey brain and relate it to other vertebrate brains. Finally, we will have an open question and answer session. So during all the discussion, if you want to participate in any of the blogs or in the final question and answer session, you can use the Ask a Question feature on Crowdcast that you can find on the bottom part of the screen. We will keep an eye on them and might invite you to join the stage during the discussion if you want to do so, of course. And well, with that being said, just thank our panelists, Worldwide Neuro, and I leave the stage to Manuel.
Hi, can you hear me? Is there any noise? Maria, you were saying that there was... Is it okay? Okay. I will share my screen.
There is still noise, but I guess that's what we
Yeah,
have to...
I don't know how to fix that. I hope you can hear me well. Let me just share my screen, my presentation. Okay, so here's my presentation. And your screen sharing. Oh, okay. Okay, let me see. Sorry, it's the first time I actually share this. Okay. So can you confirm that you can see the whole slide? I cannot see you, so you will need to... Yeah? Can you see the whole slide? Can you see when I move?
Yes, it looks good.
Yeah. Okay. Yeah, so as Alex said, I will try now to summarize the main ideas of Chapter 2, the origin of vertebrates and invertebrate chordates and cyclostomes. This is a very long chapter with many details, so I will have to skip some in the interest of time, and I will go fast over some ideas because, as Alex said, Maria, Paul, and Luis will discuss them more in depth later. I will, however, want to start by telling you all what I do in my research, and I will explain in a second why I believe this is relevant. My main aim in my research is to understand how rats make decisions in laboratory tasks like this one. This is a 2 alternative first choice task in which rats in this particular task, they can use their trial history, their choices, the outcomes, and I want to understand how rats use that trial history to make informed decisions. Further, I model their behavior with machine learning techniques like mainly recurrent neural networks, which allows me to generate hypotheses about how the brain of the rat is producing the behavior we observe. So this is a very broad view of my research. Of course, I guess some people in the audience might do similar things, but the reason I'm telling you about this is twofold. First, because to let you know that I'm not an expert in the things we are discussing, so please feel free to comment or add anything you consider relevant. I was learning this morning how to pronounce some of the things I will say today. So if I'm sure some of you know much more about what I will be telling. And a follow-up question is then, if this is so different from what you do is why you are interested in the evolution of the brain. This is a question that actually Luis asked me a couple of times. Why you computational neuroscientists want to learn about tunicates and lampreys? And I believe this is a very relevant question, and it's a good exercise to articulate the reasons. I encourage you to share your reasons for wanting to learn about the brain. In my case, I could think of two. The first one is, I'm not the first to say this, but I believe that many features in the brain of a rat will be very difficult to understand if we don't take into account when they evolve, how they evolve, and why they evolve. And the second reason is that if I want to build an intelligent agent, if we can call this agent, it seems like a good idea to look at how evolution created the most intelligent agents that we know, that are animals and their brains. So I like a lot when the book looks at talks about the innovations and tricks that evolution used to improve the performance of the animals. So yeah, please share your reasons if you want. In the meantime, I will move on because we have a lot of things to discuss. We will start with the first section of the chapter, in which I will not stop much because Maria will tell us more in depth. I will just say that neurons and nervous systems emerged several times throughout history, which is a clear example of convergent evolution, and tell us a lot about the importance of this particular type of cells and this particular type of organization, which I find fascinating. This section, however, is very, very separate from the rest of the sections, because already in section 2.2 we meet the basal vertebrates and their closest invertebrate relatives. These are all chordates that emerge in these periods, the Ediacaran, I hope I pronounced it properly, and Cambrian periods. We will be talking about 4 species, starting by the amphioxus. The amphioxus is a very small animal that lives in the water and has this shape with the mouth on the left and an otochord that is characteristic of all chordates, and has a relatively simple brain, although Luis will tell us more about the structure of this brain. I will just say that the brain goes from, it's a long tube that goes from the mouth to the tip of the tail. When we move to tunicates, specifically ascidians, it is kind of a surprise to know that tunicates are our closest invertebrate relatives when we look at their shape in adult life. But when we look at their larval state, it makes a bit more sense, although they are not very similar to us, of course. But they are a bit more similar to amphioxus, with this long tail and the eye, and a very small brain with 150 neurons separated in a sensory vesicle and a visceral ganglia and kind of a spinal cord. So then the animal becomes adult and it maintains the brain, I believe, to be able to feed. And I have this scheme in which you can see that the water would enter through the oral siphon and go out through the atrial siphon. Okay, so the invertebrate animals. Then we move to the vertebrate animals, but a particular group that are the cyclostom, in which we find the lamprey. The lamprey is a much bigger animal that has a shape that is more like more fish-like, if you want, with 2 eyes, with gills, with a mouth that is center and the fins, and a much larger brain with all main structures that we can find in mammals. Hagfish have also a large brain, although they don't have eyes. because they are very functionalized because they live in the deep ocean floor. These two animals are the cyclostomes. They are known as jawless vertebrates, and the reason they are called cyclostomes, if I understand correctly, is that they have this circular mouth that allows them, in the case of lamprey, allow them to latch onto animals because they are parasitic animals and suck their meat or their blood. And you can see also the mouth of hagfish that they close laterally. So the book emphasizes that this was a very important innovation that allowed these cyclostomes to thrive because they could eat more. They could acquire more energy. Okay, so these are our 4 species summarized here. The book then moves to explain how this period, or what were the conditions in the Ediacaran and the Cambrian periods. And they have these beautiful images from pictures in which they show some of the animals that might have lived in these periods. During this period, temperature and oxygen increased, which allowed the animals to thrive, these small aquatic animals. And I have this sentence from Metazoa, the book by Peter Gottfried Smith, that I think it is very, it summarizes well what they say in the chapter. Early chordates and early vertebrates had to get smarter, or at least improve their detection systems and their capacity to move, to escape from predators, because although most of animals were filter feeders, they were already 25, well, maybe less, but a significant quantity of predators that could actually eat you. So you needed to escape from them. And the strategies that early vertebrates use go from getting bigger, which is a very simple strategy, but it seems it worked. They provide this example in comparing amphioxus and frogs. I have this other in which I put the amphioxus here at scale with the lampreys. And then they develop other capacities. The first thing that has to come with this getting bigger is these yields, because when the animal gets bigger, breathing through the skin is no longer efficient enough, and so gills allow these animals to get more oxygen. They got other ways of eating, as we have seen. They improve their detection systems, and as we will see in a second. Their capacity to move, they improve their locomotion, and they also develop muscles to pump water, so they could feed better. And I believe this also helped them breathe better, to get more oxygen. So all these changes require a new developmental plan, and this was helped by the fact that the genome duplicated twice from chordates to jaw vertebrates. So it duplicated here, and it duplicated again here. And what they say in the book is that this allow the vertebrates to explore or to get more complex morphologically, because they could explore other opportunities. Okay, let me stop here. Yeah. So then we move to the next section, in which the chapter discusses much in detail the different sense organs of the early vertebrates. I will just go through some of the examples that they discuss, and then summarize all the ones that are in the section. First, they mention the photoreceptor system of amphioxus that is very simple. Amphioxus only have one small eye, and that is mainly used for detecting ambient light, and also maybe for detecting predators that are approaching. So this is a huge or a big shade that is getting bigger, and so they can escape. But as I said, this is a very simple system if we compare it to to vertebrates. And then, so for instance, lampreys, well, let me say that hagfishes don't have a very developed visual system as far as I understood, because they live in very dark environments, but lampreys have already 2 eyes with larger retinas, with lens, with more cellular neurons like amacrines or bipolar cells, and they also develop these photoreceptors that are all their photoreceptors are cone or similar to the cone photoreceptors that we find in mammals. But I find very interesting that what they say that is so these photoreceptors can come in different lengths, and the ones that are shorter have a different profile of response profile, and they have a response profile that is more like rod-like photoreceptor, which allows them to perceive more information in dark environments. And they say they provide this idea of the duplex retina, because this variability in the length of the receptors allow animals to process information in different channels, if you want, which I think is an interesting innovation. I will mention also the lateral line system, because it's very relevant for many, for all fishes, as far as I understood. In vertebrates, there was the innovation of this neuromass, that is all these cells together that improve the capacity of detecting vibration in water, and they also mentioned the vestibular apparatus in which the vertebrates evolve this semicircular canal that allows vertebrates to perceive head rotations. So this is the summary they provided. This is table 2.1. They provided at the beginning of the section, and I just mentioned some of them. If you are interested, you can go over the section and look at it. I really encourage you to do so. But in the meantime, let's move to the next section, in which we will discuss briefly the movements of how vertebrates improve in their movements and their motor control. So I have this video. I hope you can see it. I don't know if you will be able to hear, but the basic idea is that you see this lateral undulation. That is how lampreys move. And although what they say is that vertebrates didn't invent any new form of locomotion, they improve it. And if you look at how amphioxus move in a video, I didn't add it here, you will see that lampreys seem to be more in control, if you want. There are other innovations that vertebrates introduced. Pumping water, I already said it, which help breathing and help them also obtain more energy. More efficient swimming, I just said, and eye movements, which probably allow them to detect predators and detect prey as well. Okay, so finally we move to the early vertebrate brains. We have been talking about these different species and the habitats and their senses. What I did here, for me, it was a bit difficult at the beginning, because there are many different areas, and the summary they provided at the beginning of the section was very useful for me, and I thought that it could be useful for other people that are like me, computational neuroscientists or machine learning people. So let me provide you with, this is the brain of mammal embryo, or the mammal embryo brain. Yeah. And so they go over the different areas that are, the main areas would be the hindbrain, the midbrain, and the forebrain. And they say what they give rise to. So for instance, the hindbrain gave rise to the cerebellum and other structures that I omit here. The midbrain gives rise to the optic tectum and the colliculus. And the forebrain is divided into the diencephalon, that give rise to the pretectum and the thalamus, and the secondary prosencephalon that contains many of the, well, at least for me, the most known structures in the brain. First the preoptic area and the hypothalamus, and then the telencephalon that includes subpallion and pallion, where you can see the striatum, you can see the neocortex, the hippocampus. So these are the areas that I usually hear of, actually. So this will be the brain, or how the brain of mammals look like or develops during the development, during the embryonal stages. Let's look at how this looks, or how this this looks for invertebrate chordates, meaning amphioxus and tunicates. So in this case, if you remember, these were their brains, very simple. So we don't have a cerebellum, we don't have a midbrain, we don't have a telencephalon. It is maybe Luis has other opinion. What I'm saying is what the information they provide in the chapter, and they don't. I believe they don't mention preoptic area and hypothalamus. So they do have a forebrain, a hindbrain, and the encephalon, a secondary prosencephalon, but the brain, of course, is much simpler. If we look at the cyclostomes, and I will focus on the lampreys, which Paul will tell us much more about later, the brain is much more evolved, but they don't have a cerebellum. As far as I understood, lampreys or hagfish don't have a cerebellum. And then when we look at the telencephalon, then things become a bit more complicated. So here you have to separate pallium. What they do is separate pallium into medial, dorsal, and lateral. And there is some controversy about whether lampreys have a medial or dorsal or lateral pallium. But again, Paul will explain this in much more detail. So I'm almost done. They have a section about the developmental mechanism of evolving a new head, in which they describe the neural crest and the placode and the importance for developing this new head. I will skip this section. I will just provide this figure from the paper they cite that I found interesting. So their idea back then was that the start of the new head was, I believe this is when chordates emerge, and then you can see the different stages here as we approach to jaw vertebrates. But in the interest of time, I believe I'm a bit over time, I will move to last section of the chapter, in which they go back to the question of novelty. They touched on this in the first chapter, and we discussed about it in the first session. What they said in this section is that when one wants to discuss about or to debate, when there is a controversy about whether a feature is new or is homolog to or to another structure, it is important to define in which level we are discussing, because we can have new genes that are expressing all morphologies or morphological structure, or new morphological structures that are expressing, that are produced with all genes, and the same can be true for behavior. So basically defining exactly what is the level at which we want to define, or at which the character is making its function is very relevant. And they add this sentence that is more a question: how much change do we allow before we call a structure new? And then they cite Jacob, which says that to create is to recombine, which I find interesting. So yeah, I'm done. We will now move to the discussion, in which we have the different sections for Maria, Luis, and Paul, and then we will have the questions. So we will be 5 minutes and then 15 minutes of questions. So please start adding your questions regarding these topics in the so far, and then we will see whether we move to other topics. We'll stop sharing my screen. I hope it was clear. Yeah. So Maria, do you want to share your screen?
Okay, so I can start right away. Let's see. This works. Can you see my screen? Can you hear me well? Can anyone give me feedback?
Yes.
Okay.
Yes.
Because I don't see anything now.
I see a black screen right now. I don't know.
Ooh. Okay. This is not what I meant to share. Let me try again. Is this better?
Yes.
Okay.
Yes.
Can you see just the slide and nothing else?
Just a slide. Yeah.
Okay. Wonderful.
Yep, looks great.
So the purpose of this is just to introduce some bullet points to some interesting discussion points. But this is really what we will talk about later is really something that we want you to drive, and so it will be connected to your interests and to the questions that will come from you. So I'm just going to expand a little bit on what the chapter says on the evolution of the early evolution of neurons and nervous systems. And what you see here is a phylogenetic tree with, that shows the relationships between vertebrates and invertebrates. And in red, what I think are still the big open questions on brain evolution, nervous system evolution. And there's been a lot of progress in the last 5 to 10 years in this field. The progress has been driven by new technologies. First of all, the sequencing of genomes, and now single-cell RNA-seq, and even some whole animal connectomics for small invertebrates. And there's a lot that we learned in the past few years on the evolution of the first neurons and then the evolution of the first brains and of the vertebrate brain. But as it will certainly come up during the discussion, there is still a lot to learn. And these big questions are still open for a number of reasons. One is the phylogeny. As you can see in this phylogenetic tree, some of the most important animals that people are studying to understand the early evolution of nervous systems, and for example, the comb jellies or ctenophores that are indicated here, or sponges, they still have unclear placement in the tree of life. For example, for ctenophores, it is unclear whether they are the sister group of all the other animals of cnidarians and bilaterians. Bilaterians are these animals here that have a bilateral symmetry at least in some stage of their life cycle. And cnidarians are animals like jellyfish or sea anemone. And all these animals that you see depicted here, they have a relatively clear phylogenetic placement, at least when you go to the major clades, and they all have nervous systems. But then when you start looking at ctenophores or sponges or placozoans, which are some other basal metazoans, it is unclear whether their placement in the tree is unclear, and this has consequences on whether whether neurons, for example, on the conclusions on the evolution of neurons, whether neurons appeared only once or multiple times in evolution. So I'm really talking about animals that are not so common to, especially to people that are doing systems neuroscience work. But I want to highlight some of the lessons that are coming up from applying neuroscience approaches to the study of these animals, because I think there are some major implications for understanding nervous systems and the brain that come from this work. Can you still see my presentation? Something is happening here.
I can see it.
Yeah, it looks great.
Yeah.
Now,
Oops, now
I can.
I apologize. Okay. Now it's back to normal, I hope. So for
not yet.
No?
Not yet.
No.
No, still did not come back.
Is it telling you?
It was fine though.
Now it's coming.
There.
Now it's black.
Yeah. Oh. This is so annoying. Okay, now should work.
Yes.
You see it?
Yes.
Okay, excellent.
Very good.
So, I don't want to highlight so much questions on homology, but rather questions on function, because I know that many of you are interested in brain function. And I think there are some very important lessons to learn from these animals that either don't have nervous systems as we normally define them, or that have very simple and very simple nervous systems. So the first thing I wanted to mention is that there are some basal metazoans that are called placozoans, and you see here represented in this schematic, that that don't have true neurons. So the definition of neuron in this particular case is of a cell that is excitable and that establishes synapses with other neurons. So there is nothing like that in these placozoans. However, recent work has shown that these animals have a very complex neuropeptide repertoire, and they do use neuropeptidergic signaling for what we can start calling behavior, so for controlling their locomotion. And in these animals, like in many other basal metazoans and even bilaterian larvae, for example, locomotion is not based on muscles, but is based on cilia. So you see here in this schematic, there are these ciliated cells, and the activity of sensory cells and neuropeptides modulates the beating of these cilia and basically controls how the animal moves around. So what you see in these videos are some of these placozoans that were treated with different neuropeptides. These are neuropeptides that have been found in the genome of these animals that have been synthesized, and you see how they react differently to the application of these neuropeptides. I'm going to play this again because it's just very nice to see how the same animal, even without a nervous system, is showing these very distinctive reactions and behaviors to the peptidergic signaling. So one of the lessons we're learning from all this work on basal metazoans, like these animals, is that peptidergic signaling had an ancestral role in animal behavior, probably predated the evolution of true neurons. And I think this also raises interesting questions on what are all these neuropeptides doing in more complex brains, such as the mammalian brain, which has a very high complex repertoire of neuropeptides and GPCRs, but this somehow doesn't really find enough space in the current work in systems neuroscience. And another thing I want to show you, now we're moving to cnidarians, so to animals like jellyfish and sea anemone, is how a nerve net looks like. Because it's thought that the earliest stage of nervous system evolution, once neurons evolved, was not a centralized brain, but a diffuse nerve net. And this is what I'm going to show you here is a footage of Hydra that is a polyp that and this animal is a transgenic animal expressing GCAMP in every neuron of the nerve net. And so what you will see is that this animal has different patterns of contraction and relaxation of its muscles, and these patterns of contractions and relaxations correspond to the activity of different subsets of neurons in this nerve net. So initially, it was believed or thought that nerve nets were kind of homogeneous, but this kind of functional work, together with transcriptomics work, is showing that actually nerve nets in cnidarians are regionalized, and there are specialized types and subtypes of neurons within these nerve nets that have different roles in behavior, which I think it's very interesting, and there's also some implications on how we think about the evolution of true centralized brains from a diffuse nerve net. And another bullet point that I want to put out there is, and now we're moving to bilaterians, is one of the big questions that it's still relatively open is what is the relationship between complex brain centers in in vertebrates, and brain centers like the cerebral cortex that are implicated in learning and memory in vertebrates. And I think many people would assume or believe, and this is also the point, the position taken by the book, that these high brain center, sensory associative nerves brain centers evolved convergently, and this is definitely what the phylogenetic distribution of these brain areas suggests. If this is convergent evolution, is very interesting to think about the fact that the genes that pattern these brain areas, if you look, for example, at the mushroom body of an insect or an annelid, and compare that to the cerebral cortex of mammals, of vertebrates, the genes that are involved in patterning these brain areas are the same, and they're expressed in similar topographical relationships. And also some of the computations that are executed by these brain areas are comparable. There are many aspects that are very similar. For example, the fact that there is a dopaminergic system that is impinging on these brain areas and is involved in associative learning. So there are shared principles, and if this is convergent evolution, it would be very interesting to understand more in detail what constraints from a molecular, developmental, or functional perspective enabled the evolution of these structures that share a few surprising similarities. And then a final point that I want to raise is that the reason why understanding the evolution of the vertebrate brain is, in particular the vertebrate bow plan, as we will talk more about later, is so complex, is because the closest relatives of vertebrates are animals, deuterostomes with relatively simple nervous systems, and that also have some adaptations in their lifestyle and in their body plan that may in part reflect secondary simplifications or divergence from a common ancestor. One of the most interesting findings of the recent years is that when you look at hemichordates, which are deuterostomes that closest relatives to chordates, so to the tudicates and the amphioxus that Manuel was talking about earlier, is that they also have a relatively diffuse nervous system. Some people have been talking about this as a nerve net. It's not exactly a nerve net, but it's a very diffuse epithelial nerve plexus, at least in the anterior part. But the genes that pattern these nervous systems are expressed in a similar way as the genes, transcription factors, and signaling centers that pattern the vertebrate brain. So in these animals, people have identified things like zona limitans intrathalamica that patterns the thalamus and the forebrain, or other I don't want to go into specific names, but it's these key signaling systems that regionalize the brain. So it's one of the open questions in the field is whether there is more conservation at the level of brain patterning and less conservation at the level of cell types, or whether there's just a hidden level of complexity in terms of cell types that still needs to be uncovered. And I will stop here because I really want to go into the questions of the audience.
Yeah, so we have a question, but I don't know if Luis or Paul want to make any question or comment. Paul, could you turn on your camera because we cannot see you?
My camera?
No, Paul, I see you.
Oh, I think it's on. I
don't see him.
Oh,
okay. So
how strange.
Me.
Yeah. Anyway, well, I had one question, but I'm going to let it go it's similar to what the person was asking, Malcolm, on the thing. So I'll let that one go. What I really wanted to discuss a little bit about this idea of brains, whether there's a centralized brain etc. And this idea that there's a nerve net on one end, and that's sort of not really specialized, and then there's a very highly specialized brain that you can find in vertebrates, etc. And the thing in hemichordates is a little bit like in between those things, and maybe the hydra is also not quite unspecialized. And it seems to me there's a lot of focus on sort of these kind of structural organization, but the functional organization seems to be there anyway, whether the structural organization is clear or not. And this is one of those debates that I know Maria has been involved in through her work in Detlef Arendt, and so I wanna get you talking about it. Essentially, Arendt, quite notoriously, I think, has suggested that the fundamental sort of organization is quite shared among a very large body of animals. Essentially, all bilaterians have this kind of a merged, merged regions of the ancestral neural net, the apical and blastoporal nervous system. And at the site where these two things merge is where you get brains emerging. And so there's a sort of a very, very deep origin of what is a brain. Whereas Northcutt, in his own writing in response to that work, as well as in the book, suggests that, well, no, real brains don't really, you see them really scattered through the tree. They have a picture in the book of where you have, Manuel had it, I think, in his presentation, where you actually get a brain. And, chordates have it, insects have it, certain mollusks have it, etc. But many other species do not. And so I just wanted you to comment, because I don't know really, how important is it, that kind of centralization itself, and where do you, now, you may have a particular stake in the debate, but I'd like to hear you and you discuss it and sort of reply to some of the questions that have been raised.
So one of the things I should say is that I've, as you mentioned, I've worked on these topics as a graduate student, and that's been a while. So I'm since 2014 I started working on the vertebrate brain and got more interested in cerebral cortex evolution. And I must confess I didn't keep super up to date with the literature. I mean, I followed the basics, but I think the part of the controversy arises from the fact that many people that come into this field, they come from a very classical training in morphology and zoology, where things like the presence of a certain number of nerve cords, or these very tangible morphological characteristics, were the main characters that people were studying to infer phylogenies and evolutionary history. And when you start overlaying on that level of description the information we're gathering from expression of genes that regionalize the nervous system, things get more complicated, because sometimes you don't have an immediate a clear-cut relationship between the expression of a certain patterning system and the morphological structure. And the hemichordate example that I brought up is a very good example of this, because the proboscis of the hemichordate has this very diffuse nerve net, essentially something that looks like a nerve net, or very diffuse basiepithelial nervous system, but you still have conservation of patterning. So I think there is this disconnect between talking about patterning and talking about morphology, and sometimes people they think they're talking about the same thing, but they're actually talking about different things. What will be rewarding in the future is to look at cell types, because neuron types should be more comparable than, let's say, the number of nerve cords. And I think I would pretty much be convinced by seeing the same cell type if we see the same cell type repertoire, but in one animal it's more condensed and in another one is more diffuse, I will still be be pretty much convinced of a common ancestry of the cell type in that case.
What about physiology? If somebody went through and actually did experiments, let's say during the locomotion of a basal protostome or something, trying to understand the physiology of these cells, do you think that we would actually see, like you do in the genetic patterning, evidence for very old circuits, functionally definable things
that would be
old?
But I will not bet on that. The problem with physiology, in the same way as we see when we look at the vertebrate brain, is that you can have many examples. There are many examples of convergent evolution of function.
Mm-hmm.
But I think a good place, it would be, for example, very nice to get a deeper understanding of motor neurons across all these animals, including cnidarians and other metazoans, because one of the predictions of the chimeric brain hypothesis that you just mentioned, the idea that there is a blastoporal nervous system and an apical nervous system that trace back to the last common ancestor of cnidarians and bilaterians. One of the implications is that some of the neurons that control certain types of muscles in cnidarians should have a common origin with motor neurons in bilaterian animals. So that's something that needs to be addressed more thoroughly at the molecular level, but also the functional level. I think that would be very interesting.
Very cool, thanks.
Yes, I guess we can invite, unless Luis wants to say something, I guess we can invite Malcolm McIver to the stage. This has to be done by Raquel. Yeah, there is some delay. Yeah, so disconnecting.
Where is the screen? Where's my screen gone? Can you hear me okay?
Yes?
Yes.
Oh, hi Maria. How are you?
Hi. Good, how are you?
Yeah, so fascinating point you made about peptidergic signaling, and I wondered if you knew, with respect to cephalochordates and tunicates, whether there's more reliance on peptidergic signaling there. And I guess what I'm thinking is, if you think about, the basic point of a cell having an insulating membrane, and therefore excitability is easily sequestered, easily channeled within a cell, whereas peptides are kind of messy and spread, and it's a little hard to control. That maybe it's necessary, as you get more complex, to go to modes of information flow that are more easily controlled. And I guess the test would be, as you look at intermediate forms, would you see a diminishment kind of smoothly as the animal goes up in complexity, as animals go up in complexity? Is there a smooth diminishment in reliance on peptidergic signaling?
That's an interesting question. I don't know in detail the situation for tunicates and cephalochordates. I mean, I know they do have lots of peptides, and they're expressed with some cell type specificity. The way I'm thinking about this is very influenced by the work of Yves Marder and Cory Bergman, for example, that they argued for these two orthogonal levels of function. The synaptic level and then the peptidergic level as an orthogonal level to control brain state. And it might be that this more basic brain state kind of peptidergic control is more ancestral. And how the two systems interacted in evolution, I think it's at this point not very clear. But I can imagine that things like, as the same principles that have been elucidated in the work on crabs or on C. elegans or other species where the interaction of these two systems has been worked out, I can imagine that would apply also to, let's say, early bacterial nervous systems. I don't know, Paul.
I thought that was very interesting that you mentioned that work from Yves Marder, because it does look like, but it does look like even in modern, very specialized brains like ours, we still have these peptides, and the regions that really sort of traffic in those are like the hypothalamus and some of these neuroendocrine systems that have this widespread untargeted modulatory effect, and so perhaps there are still those things there in our brains and others. It's just that the set of other circuits, those synaptically organized, very specific circuits, has just grown so much. And maybe the balance has shifted not because peptides were just not that useful, but because you just build a larger system on top of those peptides, always using this kind of modulatory role. But I don't know. I mean, I think it'd be interesting to do some of those experiments that Yves Marder has done on vertebrate branches.
Exactly. I mean, look at for example, I'm biased here because I look at the GABAergic interneurons in the cortex, and they are defined by their expression of different sets of peptides, and some of these peptides are conserved in conserved cell types, at least within amnions. So one wonders why there is so much. There are the specialized peptidergic systems in the cortex where we don't think about it. And one possibility is a modulation of neuronal activity at medium to long time scale, because peptides act at those time scales. They're not, at least to my understanding, they're not acting on the millisecond time scale as action potential. Now, so maybe these peptidergic systems integrate information over a long time and modulate the brain state over these longer time scales. But
okay, yeah, that's helpful. Thank you. Well, I have you one other quick question. It's kind of a crude question on mass. So I kept feeling from chapter 2 and looking at what you said about placozoans and the book as well, that simply by increasing size, an animal has all of these additional demands, such as you can no longer rely on diffusion of oxygen through the skin and so on. So now you need pharyngeal slits that are pumped with water, et cetera, et cetera. And it just gets very complicated as you add mass. And so my crude question is, have you or anyone else that you know of looked at the simple question of, if you have on the y-axis mass in grams and on the x-axis number of cell types, be they neuronal or more generally, is there a relationship there? Or is that way too crude?
I never looked at that. That's I would suspect it's not as simple.
Yeah.
There's a lot of cell type diversity, even in small brains and simple animals.
Okay.
It must certainly come into play when you think about locomotion and the kind of systems you need to evolve to control locomotion, big animal versus a small animal. Efficiency also of information transmission. Maybe there is a relationship between the evolution of true action potentials and the bigger animals. These are some of the things I can think of. I don't know if Luis or Paul want to add anything.
I don't have much to add. I think it'd be great to see it and then see what it would look like, frankly. But
Yeah.
Okay, thank you.
Thank you, Malcolm.
Jump off the stage for now.
Thank you, Malcolm. So I believe we have to move to the next section with Luis, because otherwise we are running out of time. So Luis, if you want to share your screen. You are muted now. I think I cannot hear you. Can you hear
Do
me?
you need to refresh my browser?
I don't. Do you need to? I don't see any screen, so I don't know if you're trying to share.
Let me wait a second. Let me reset here. I'm trying to share now again,
I will ask you if
but I get a message whether I want to share to Twitter, Facebook, or LinkedIn. There's nothing like that, huh?
no. No, it's you
So I
should
don't know why I get that.
hover. So you know how to share? You have to hover your pointer over your screen.
Yeah. Let me see. Here, let me see.
You can share yours. Do you have many slides? Because
Only 2 slides. Only
Okay,
two
so-
slides.
So then I can because you send them to me. so
I seem to have problem with getting this to work.
Mhm.
I sort of sometimes transiently I see something, then it goes out. Here it is. Let me see whether I get it now. Okay, there we are. Okay, do you see it now?
No.
No, it's full screen for me now.
Yeah. But probably you are not for some reason you are not sharing it.
This is the second slide. I'm seeing now the second slide.
No, we don't see your slides.
No.
Yeah. So
Yeah, let me see. Seems to be sharing something else.
No.
Then please put my presentation from your side. There's only
Yeah.
2 slides.
Paul, could you? I just send you Luis' presentation because I cannot open.
Oh, okay.
Properly.
All right. Let me see.
Yeah.
Let me see. I can.
Did you receive an email?
Here,
let me
here is something that I should restart my computer. Should I go away and reset it?
No, please don't. We will share your.
Okay. I think I have it. All right. Let me just put it somewhere.
Maybe you can present my slides.
Yeah, I can do the slides. Yeah, I just won't be able to know where you want to point. Okay, so here we go. Book Club Amphioxus. And
Or I can speak if you can see it, but I don't see it either. I only see your faces.
no, I'll share it in just a moment. It's just bringing it up still. Oh boy. Okay, there we go. All right, so I'm going to share screen now. Okay. Okay, can you see it?
No.
Nope. Okay.
It says I'm sharing the entire screen.
So do we have an issue with this sharing because we are all having?
Oh yes, I see. The little button is spinning.
Is it trying to share it but it can't?
Yeah, it's funny because
now-
we did
now
do
we
it
can
before.
see
Oh,
it.
here we go.
That's coming.
It's a bit slow.
Okay. There.
Okay.
Now do you see it?
Yes.
Not yet, but it seems to be coming.
Yes, I can see it.
Now I see it. Okay.
Yep. Okay.
So I can only see the upper half of the screen, not the lower one. So there's the drawing of amphioxus itself. I don't practically see it, but I know what you see there.
Really?
So let me describe what I wanted to say. We studied recently in 2017 about 80 different genes in a larval stage of amphioxus, and we chose out of that some 50 genes that were more informative. And the idea was to check whether markers telling us about the longitudinal and dorsoventral subdivision of the brain in vertebrates, which generally follows what we call the neuromeric or prosomeric model, according to our data, whether some trace of this sort of subdivision bow plan was to be found also in the amphioxus forebrain vesicle at larval stage. In the upper higher right corner, you see some schemata, and surprisingly we found a lot of evidence for commonality in the basic fundamental pattern, although of course there are big differences as well. For instance, there is a sort of primary separation into archencephalic and deuteroencephalic, which is more or less decided by the boundary at the level of the isthmus, where you are going to have the isthmic organizer. That is the primary probably subdivision along the anteroposterior axis of the brain, and that you find in amphioxus as well as in all vertebrates. And the archencephalon would contain the secondary prosencephalon, the diencephalon, plus the midbrain. The midbrain will be also belong to the rosal part of the brain. And that we have evidence also for that in all vertebrates. There's a sort of commonality, particularly of dorsoventral patterning mechanisms, which are shared among midbrain diencephalon and secondary prosencephalon. On the other hand, according to dorsoventral patterning, we found strong evidence for a floor plate that was very similar to the floor plate that you find in vertebrates, ending where you would expect at the floor of the hypothalamus of the secondary prosencephalon. And there is also some evidence for an adult basal boundary, which is another important longitudinal landmark in the brain. All this because of differential gene labeling patterns that we found in the amphioxus. So in the right schema of the upper right-hand corner, you can see a flat mount of the amphioxus neural vesicle, where you can see in the center of symmetry, you see the floor plate. You can see that it does end at a particular point, not reaching the border of the neural plate or the neural tube, because that will be the roof plate, and the floor only extends up to a given point corresponding to the floor of the hypothalamus. Then you see parallel to that a subdivision of the lateral walls that goes round rostrally from side to side. That will be our basal boundary, and theoretically there should be also a roof plate, but we did not look into markers for the roof plate, so we don't know exactly how far that goes into amphioxus. Important thing is that when you now allow in the drawings to the left, you can see that in vertebrates we have in light blue a rostral territory that contains the hypothalamus extending into pieces into telencephalon. Then there is a transition into diencephalon through the prethalamus, PTH, and then we have the diencephalon, the colored diencephalon, separated by the zonal limitans organizer from prethalamus, the thalamus, and the pretectum, and then behind that the midbrain that finishes with the isthmic organizer. So all the distinct thalamus, pretectum, midbrain domain is enclosed between two organizers, important organizers, the zonal limitans, which is the interthalamic boundary, and the isthmic organizer behind. Behind that, you find the hindbrain spinal cord, and when you look into the amphioxus, the lower drawing, you can see that more or less the genes show that the same general subdivisions, with the difference that the entire set of thalamus, pretectum, midbrain in amphioxus is reduced to a very small domain. In the larval stage where we look at it, it was reduced to 4 cells at the right and 4 cells at the left side. So it was a very tiny domain that had no particular genes that you could compare with either thalamus or pretectum or midbrain, so it has a sort of completely undistinct molecular profile, so we don't know what it really means, this amphioxus the MES territory, but it's in the place of the caudal encephalon and the midbrain. So it's clearly bounded caudally, but it could be a sort of isthmic boundary. But as that isthmic boundary, you don't have FEF8, nor do you have WID1, so you don't have the usual markers revealing that in the amphioxus you don't have the organizer itself. But you have the boundary. So the boundary itself appears before you obtain in evolution the organizer properties, apparently. That will be our reading of that. And in the case of the notochord and the floor, you can see that the extent of the notochord, this blue, light blue under the floor, you can see that it's coextensive. You get a floor plate precisely as long as you get a notochord. Of course, the notochord in amphioxus is peculiar because everybody knows that the notochord in amphioxus seems to extend rostrally beyond the forebrain into even the tip of the head. Actually, we have prepared recently a new paper where we discuss this point, and we argue against this idea, and they believe that the notochord in amphioxus also stops at the same place as all vertebrates, and the extension that seems to be a rostral notochord actually must be some sort of derivative of the precaudal plate. So we argue through some genetic data and some additional developmental. One important idea arguing that this thing cannot be a length extension of the notochord is that the floor plate is not extended beyond the mammillary, the hypothalamic floor domain, so that all this domain in front of the forebrain can, even if it has something similar to a notochord, cannot be really a notochord because the longitudinal landmarks like the floor, the alar basal boundary, and the roof domain, all of them go perpendicular into that area. So that will be in a patterning consideration that could be impossible. You cannot have a ventralizer structure like the notochord, but should induce a floor, and this floor should induce a completely different longitudinal pattern of the other structure. It could not be possible that you have this alar basal boundary or this floor plate if the notochord really extended rostrally. So that we believe that actually this shows that there cannot be a notochord in this position. It has to be precaudal plate. So moving to the next slide, please. If you move, can you move to the next slide? Here I will show some of the argument we use. We look back into gastrulation, comparing it in invertebrates with the amphioxus gastrulation and with the gastrulation in lampreys and other vertebrates. So here here you see 4 images taken from the older literature back in 1904 or so, or no, in 1894. So that here you can see under A, you can see the early stage of gastrulation, where the image you get is that the endodermal tissue prospective blastomeres, they invaginate as a hole into the blastomeric cavity, and you can see in pink what is going to be the rostral end of the endoderm, interembryonic endoderm. The rest will be other more sort of extra material. This pink domain touches what is going to be this fated to be the neural ectoderm of the amphioxus, and you can see in blue where is the material that is going to build the notochord. Is in this caudal part, caudal to the neuroectoderm, and from that area you can see there are green arrows going out both at the dorsal lip of the blastopore and below at the lower lip of the blastopore, and that tells you what is the direction of growth of the embryo. These are the area containing the most intense mitotic activity, and that is where both the brain and the skin and the gut and every part of the embryo, they are all actively growing caudal walls, where other more rostral positions are not practically growing. So that as you compare with B, you can see that now the length of the entire head of the embryo has increased. Now your blue notochordal material has entered also gastrulation behind the endodermal pink material, and you can see in this second image how we already have this precaudal plate, this pink contact between the cephalic endoderm and the rostral part of the neural ectoderm. So this is the primary precaudal plate, and you really can see that because it's there beforehand in A, that means that the notochord cannot extend beyond that because there is an adhesion between endoderm and the neural ectoderm. So the notochord starts just behind the precordial plate and extends caudalwards, where it has this mitotic activity. So the notochord does not grow from caudal to rostral as the embryology books tell us. It grows from rostral to caudal as the entire head of the embryo and the entire embryo. So as you proceed, the embryo will always elongate caudalwards, and the notochord, together with the brain and with everything else, is elongating caudalwards. So in the under C drawing, you will see again in a later stage the same attachment of endodermal precordial plate with the rostral part of the neuroectoderm, and then now the notochord in blue is longer, and it is inducing at this point, the notochord is inducing already the floor plate of the future brain, which is going to end at the bottom of the hypothalamus, where you have the mammillary invertebrates, the mammillary body. So this tells you that the precordial plate is touching the brain at the prospective place where you will get the infundibulum and the hypophysis. That's a place where you get primarily this important, probably inductive effect of the rostral endoderm with the brain. Okay, we move now to the lower images. Well, here you can see in red, in the D, under D in red, you see at the point in which secondarily you get this sort of rostral extension of the apparent notochord, but you can see that this rostral extension appears precisely where we had the precordial plate before. So I think that what happens there is that there is an epithelium-mesenchymal transformation, secondary transformation of the precordial plate from an epithelial structure into mesenchymal structure, and then you lose the adhesion of the precordial plate to the neuroectoderm, and you obtain new free cells there that invertebrates will migrate across the acroterminal portion of the hypothalamus and reach the olfactory placodes and so on. And during this process of migration, that's where the eyes get divided into right and left, the encephalic vessels get divided into right and left, and the olfactory placodes get divided into right and left. So you need this secondary epithelium-mesenchymal transformation of the precordial material in order to be able to obtain all the precordial effects that you see in vertebrates. And you see that in amphioxus, everything is more or less similar, only that the precordial transformation gets transformed into a rostral extension of the notochord. That's peculiar to amphioxus, because we know from the ancient remains in the Cambrian strata of the ancient forms of amphioxus-like animals, and they did not have a rostral extension of the notochord. In the beginning, in the evolution of amphioxus-like animals, the notochord was exactly similar as in all vertebrates. In the lower images you see to the left, what is the situation in the lamprey larvae, where you can see the same as in amphioxus. You see an endodermal epithelial attachment at the level of the future tuberal infundibular region, and behind that you see the beginning of the notochord, the rostral end of the notochord. And at the right, then you see in the cheek embryo, where you can see that's an early neural tube of the cheek. I'm sorry that the letters are inverted. I don't know why this happened. And then, but you can see underneath the floor plate of the brain, you see the notochord with an end there, and below the notochord you can see the rostral end of the cat, and you can see that at this point the precordial plate is starting to detach from its previous attachment to the infundibular area of the hypothalamus. So that for me, this behavior of the notochord, that is really the starting point of the vertebrates, of the chordates, because the invertebrates never move the blue material. If they have it, maybe they don't even have the blue material, but they only do ectoderm plus endoderm, and that is basic gastrulation in any invertebrate. But the invention is to develop this new blue material. For that you need apparently so-called the new coop center, which is an organizer which acts before the primary organizer. So before that you need, and that is endoderm inducing mesoderm, prospective mesoderm. And once you get this new blue material, then this blue material can enter into gastrulation and produce a notochord. And that's when you have a chordate, when you produce a notochord. Without the notochord, you are not a chordate. Okay? And once you have a chordate, then you can become a vertebrate if this notochord interacts with the Semitic material and so on, and you produce a vertebra and so on. So in this sort of set of schemata, you see the evolution from the point of view of development between invertebrates into amphioxus, which is the cephalochordates, the most primitive or basal cephalochordates. From there you will go into tunicates, which have the same sort of restoration as you see in amphioxus, and from there you move into lampreys, into all vertebrates. So that is the general idea. In what sense an amphioxus or chordate, primitive chordate, is similar or non-similar to the brain of a vertebrate. I would say that in several important aspects it's slightly similar, but it's mainly non-similar in this strange dimesencephalic initial intermediate part. Functionally, the hindbrain spinal portion of the brain probably is active in reflex action and probably integrating different sensory inputs into reflex motor activity. It's not clear what this intermediate blue domain is going to do in the amphioxus. Nobody has any idea about that, and there are little data about connections out of this domain in the adult. And the hypothalamus is the most similar structure molecularly with vertebrates. So that means that you don't have a telencephalon, you have only a median eye primordium, there are no bilateral eyes, but you don't have also something on the medial plane, rostral-median plane, that could be a sort of hypophysis, beginning of an hypophysis, median eminence territory. So there might be a neurohumoral effects coming out of that hypothalamus. There may be some visual sensitivity, and surely the hypothalamus is one of the first functional parts of the brain that you would postulate in order to maintain an animal alive. So that means that in these animals, pending the solution of the mystery of this intermediate thalamoprotector mesencephalic domain that is not there yet, you seem to have neurohumoral mechanism devoid of a telencephalon in the rostral part of the brain vesicle. Of course, if there's no telencephalon, you don't need a thalamus, and then all these pathways going through the thalamus to telencephalon don't exist in amphioxus. All this is beyond the level of the chordates, and this only appears in vertebrates. But you can see that the general longitudinal and dorsoventral pattern of the brain is already there. We already have a rostral end of the brain that receives very primordial messages from the endoderm, not from the notochord. The notochord affects little the hypothalamus, I suspect. So it's mainly the endoderm, the early endodermal, which is what we share in common with the invertebrates, what patterns probably the beginning of the hypothalamus and the prospective telencephalon, which the telencephalon, after all, is for me a derivative of the hypothalamus. So it's just a dorsal domain of the hypothalamus that becomes large and enlarges and evaginates and so on, similarly as the eyes. So that is just secondary. So the question is whether in amphioxus there's any functional connection between the hypothalamus and the hindbrain, for instance. It would be nice to find maybe there are some neurons in the hypothalamus that connect with the hindbrain, and that will lead to some sort of unified functioning of this animal, where what happens in the rostral part relates to what happens caudally, but probably the central part is not very functional. My guess is that it's waiting to be induced by the introduction of the zonal limitans and by the introduction in evolution of the isthmic organizer, and once these two things appear, then you develop the thalamus protectum and midbrain. We checked that in our initial paper. We looked at several fish and mouse, double mutants, lacking both the isthmic and the zonal limitans.
Luis,
I'm finishing. What I suggest, let me finish this sentence. When you take out the two organizers in mouse and in several fish, you reduce also the central portion of the brain. Okay. Finish.
Thank you. Because we have several questions from different participants. So
yeah, actually, I wanted to add something functional to this, because I know a lot of people are interested in function. So a lot of my understanding of amphioxus comes from reading the work of Thurston Lacali, whose stuff I like because he's not afraid to speculate, and I want people to speculate about function. And one of the things he proposes actually is that midbrain region, what lands in your dimesencephalic region, includes in an alar structure that he suggests is a tectum, a little bit like a pretectum maybe, rather than a tectum specifically. And the work, and basically based on its projections. And yeah, so I'd like to hear your opinions on this.
Luis, you are
there is no recognizable midbrain territory from the molecular point of view. No thalamic, no protector, and no midbrain. Nothing. Absolutely no gene coincides. You don't recognize any midbrain at all, molecularly. No things, no PAC6, nothing. No markers there. So
Yeah,
I cannot say that.
But the connections and the
Yeah,
development
but the connection in homology doesn't work very much. If you don't can homologize the territories in a bauplan, then having a connection doesn't mean anything. Anything can connect with another thing. That's my opinion.
I will just structure that part saying that having a connection doesn't mean anything. If the connection from the frontal eye to this territory, in your opinion, is not homologous to the connection of the eyes to the tacting, then it needs to be explained in some other way. It needs an explanation. So what's your hypothesis?
There was that Vopalensky paper that suggests.
Yeah, I mean, you
etc.
You can have a particular connection doing some important function. But if you want to homologize, you have to homologize both the beginning of the connection and the end of the connection. If you cannot prove that this is a midbrain, this is a spinal cord, then you cannot talk about tectospinal connection. You can talk of any other name. You put the name and the signification to the connection. But you cannot homologize. You cannot say this is tectospinal because you have not identified, in this case, the tectum. You cannot say that the tectum, this area is a tectum. If the genes don't coincide, you don't have evidence for that. Just having a neuron there, well, does it mean that you have a neuron connected to a spinal cord? You don't know why this neuron connects with the spinal cord. It can be anything. Pretectum also connects with the spinal cord, and prethalamus also connects with spinal cord, and the encephalon connects with spinal cord. So which of these things is this connection? If you don't identify
the story is that it's an escape circuit.
You have to identify the territories. That's the first step. If you don't identify the territories, then you are in analogy. You are not in homology. You are doing this thing, seems to be doing the same function as this other animal, but with different territories. Okay, analogy. This is analogy. Analogy is also an important process in evolution. Analogy, but not homology.
Sorry, we have a delay.
I mean,
so they even bring up the telencephalon homologue that again Arendt recently suggested.
I would admit, for instance, that maybe in the dorsal part of the hypothalamus of amphioxus, there's an area there next to the roof plate that may be the primordium of an telencephalic area, the same as the eye patch is a primordium of the eyes.
Yeah. So it's where it should be, right? Yeah.
You may have a sort of primordial area with even primordial neurons doing primordial connections. So I admit all that. So then maybe you have some telencephalic primordial functions starting to appear already in amphioxus, and you only lack the big proliferation that leads to the vagination. Okay, that can be secondary issue, not important. So for me, the important thing is that you will prove genetically that this patch of tissue in the dorsal hypothalamus is telencephalon-like. So you will need, for me, to have PAC6, and some portion will need distalless, and you will need FOXG1 gene appearing somewhere. And once you prove that the identity of this territory can be accepted to be sort of telencephalic, then we can start continuing talking of homology, of telencephalic-like things. But not before you prove that this is telencephalic, not because just the position. Because maybe in evolution, you are not yet there. Maybe you are just a few million years before you can speak of telencephalon. We are at the beginning of the story. I don't believe that any vertebrate has a telencephalon, against what Maria sort of suggested because of some literature and so on. I think that this is postulating homology without considering bauplan. So it's just going to say, if I look at this property, then I call it telencephalon. Okay, you can, if you want.
I mean, there is an argument that the argument is based on the bauplan, is based on patterning genes. It's the problem is that the body plans are different, and so it's harder to recognize the similarities in the bauplan. But the similarities in the
how can you have a bauplan if you don't have a neural tube?
Well, because the neural tube is a vertebrate innovation, but
you don't need the neural tube in your bauplan.
For the innovation. I mean,
you need a neural tube. Otherwise, you cannot do a bauplan. You are before the bauplan of the neural tube. You are many million years before, not 1 or 2 million years, hundreds of million years before. So you cannot speak of having a background of the brain of vertebrates because you don't have in the neural, you don't have a neural plate. You don't have a neural tube. And so you don't have a telencephalon because this telencephalon is a part of the neural plate and the neural tube of vertebrates. So any time before, you may have neural features, of course, neural molecules and neurons and many peptides and many transmitters and many receptors. Any of these particulate portions of the neuroscience you certainly have in invertebrates. But you cannot say that you have in invertebrates a bow plan of the vertebrate brain because you don't. You don't have a neural plate.
I don't think that. I don't think that the absence of a neural plate in invertebrates prevents us from making these comparisons. But I think we should leave time for the audience. Just a quick question for you, Luis, because I will be here. What do you think about the sarcoglossus organizers? I mean, the hemichordate organizers. The fact that the hemichordates, which branch out, which are, the sister group of chordates, they do have these brain organizers. They do have an isthmus, they do have a zona limitans, and amphioxus doesn't. Because you were suggesting that the organizers evolved later, but these literature suggests that they're ancient and they were lost in amphioxus. So what's your opinion on this?
I didn't hear very well your question, but I wonder whether you were asking about what is these definitions in the literature about the zona limitans and isthmic organizer in non-chordates, so to speak. That's what you were asking about?
Yeah, I mean, how about what is the...
I think that what they have proved is that there are localized expression of these particular genes, which are the same genes that are used by vertebrates in these organizers. But since you don't have a brain, you cannot say that this is in a zona limitas position or in an isthmic position because you don't have isthmus and you don't have the encephalon. So you cannot homologize. You can say that this seems to be molecularly similar and this is positionally rostral to that, so that the position, rostral caudal position, is analog and you can compare as much as you want, but as long as you don't have a neural plate and neural tube, you cannot make a direct, more precise comparison. That's my opinion. So that maybe the presence of these genes and the secretion of these proteins and so on precedes the formation of the brain bow plan. So as many other molecules in the brain. So most of the molecules were invented many million years before. So maybe also the inducers and the FGF8 molecule and the sonic hedgehog and so and many other inducers, they were invented before and even expressed like the hoxes in a given order, spatial order and so on. But you simply, I don't think that you can compare insect head ganglia, nervous ganglia with parts of the brain, a vertebrate. You cannot. Independent of how many similarities in Hox gene expression you find, you cannot compare a ganglion with a part of the brain because you don't have a brain, simply because of that. There's no brain there.
So, yeah, I don't know, maybe we should move to talk about the lamprey brain. I know there are several questions, but we are running out of time and we still need to discuss about the brain of the lamprey that Paul will present. So if that's okay, I think we should move on.
Okay, I'm gonna share my screen. I'll just do the entire screen. Okay, can you see my screen?
Yes.
Okay. All right, so I just want to reiterate what we were talking about in terms of the bow plan. And I'm going to essentially start off with the prosomeric model that Luis Puelles has developed. I've got a bit of an echo. Maybe somebody needs to be muted. Somebody can be muted. Anyway, the point is that as Luis was saying, there's these fundamental signaling boundaries, the isthmic organizer and the zona limitans intrathalamica, which define essentially these three zones of the vertebrate brain: the hindbrain, this dimesencephalon, and the prosencephalon, which includes the hypothalamus, and the outgrowth of it, which is the telencephalon. So this basic structure gives us a way of comparing across different animals, and this can be applied to all vertebrates. So for example, if we want to look at mammals, here's the prosomeric mammal. You have you have, again, this huge telencephalon expanded out of the prosencephalon, the hypothalamus, and you have the thalamus, pretectum, midbrain, etc., as well as the hindbrain. You can compare that to a lamprey. So this is the lamprey model with the same nomenclature. The color scheme is different, though. So I'll just draw your attention. This dashed black line is the same as this red one. This is the alar-basal boundary that's sort of separates the sort of more ventral from the more dorsal structures. The dimesencephalon would be this part here, including the thalamus, pretectum, midbrain. And the telencephalon in the lamprey is quite a bit smaller. It's this thing here. But many of the structures are there. Now, why do we care so much about lamprey? And the reason is because, well, by modern estimates, they separate their lineage that leads to lamprey, separate from the lineage that leads to us, which is along this line here, the Sarcopterygians, separated more than 500 million years ago. And importantly, also, I think that lamprey, since about 360 million years, perhaps have not changed so much, at least in terms of their lifestyle, their predatory, parasitic kind of lifestyle, and the structure of their body. So here is a reconstruction of a fossil lamprey from 360 million years ago, and here's a modern adult river lamprey. So that suggests that they're kind of like living fossils that give us a good insight into what might have been going on back here. Now, for the rest of my talk, I'm just going to show a lot of work from the lab of Sten Grillner, his group has really worked out the lamprey in all the detail one might like. For example, the PhD thesis of Marcus Stevenson Jones from Grillner's lab looked in great detail at the structure of the tectum, its projections outward for both avoidance and approach behavior, as well as projections from the retina, the different kinds of projections for retinal ganglion cells of many different types. And this organization of the tectum in the lamprey is remarkably similar to what people find in zebrafish. So here's from the lab of Herwig Bayer, who have mapped in great detail both the anatomy and physiology of the zebrafish, both for, again, approach behavior and escape behavior. These circuits have been seen in all animals, in birds, amphibians, and mammals, even in monkeys, where the superior colliculus is the homologue of the tectum. So the circuit seems to be extremely similar across these species, suggesting common origins. Another part of the brain that seems to have been extremely well conserved is the basal ganglia, or the subpallium. I'll just go back here quickly. That's this part here, okay, of the fundamental bowel plan, okay. The subpallium is the basal ganglia, or it includes the basal ganglia as well as some other things like the septum. And again, from Grillner's lab, they have mapped and compared the lamprey basal ganglia circuitry to that found in mammals. And many of these familiar structures, like the striatum, the subthalamic nucleus, as well as homologues of the globus pallidus external and internal segments, can be found in these regions, with a very similar circuitry, both direct, indirect pathways, dopaminergic signals from the substantia nigra compacta, etc., as well as their downstream projections to various hindbrain structures like the reticulospinal tract. So again, arguing for a very deep homology of the circuits, even though they look very different, they seem to be quite the same. And now the pallium, that outer part of the telencephalon, that includes the cerebral cortex, the hippocampus, many parts of the amygdala, and other structures. And it also can be found in the lamprey. Now, the lamprey pallium is much smaller, of course, but it does have, like many vertebrates, projections downstream. So you can stimulate in the pallium to produce movements of different types, and the connections have been mapped, very similar connections as you might find in mammals, reptiles, and birds. The organization, now, of course, it's not 6-layer neocortex, it's a 3-layer structure, but it's rather similar. It also has an outer cellular layer and an inner cellular layer, a molecular layer with very few cell bodies. The balance between excitatory and inhibitory neurons is about 80% excitatory, 20% inhibitory, as in this neocortex. Some of the projection patterns are similar. The way sensory information comes to the thalamus is similar in the pallium of the lamprey. Furthermore, if you subdivide the pallium of vertebrates into distinct subregions, consistently one sees at least 4 domains: a ventral pallium. I'll show them here, embryonic stages. The ventral pallium, lateral pallium, dorsal pallium, and medial pallium. And in the dorsal pallium is what in mammals becomes the neocortex, whereas the ventral becomes piriform cortex, lateral becomes parts of the cerebrum, but also insula and claustrum, etc. Now, in the lamprey, based on these sort of topological model, you would expect the dorsal pallium to be about here, ventral being down here, etc. The medial pallium, which is the hippocampus in mammals, should be around here in lamprey. Now, again, studies from Grillner's lab, in particular the PhD work of Shreya Suryanarayana, have identified many of these regions. So, for example, there is a piriform-like part of the lamprey pallium. It's where one would expect if it's derived from this ventral pallial sector. It receives information from the olfactory system, from mitral and tufted cells, etc., as in the piriform cortex. And in where you'd expect the dorsal pallium to be, right here, this is sort of a top view of it, you have a visual area, you have a somatosensory area, and you have an area where stimulation produces movement. The somatosensory area has a specialization for a kind of a head region versus a body region, and the visual area has retinotopy. If you stimulate different parts of the retina, you get activity in different parts of cells, again, as you see in mammals. Now, it's possible that this all evolved separately in lamprey, as it did in mammals. And so, because lamprey diverged so long ago, a lot has happened along that lineage. They've had 500 million years to develop all this stuff. But again, the fossil evidence suggests they haven't changed that much. And so, on one hand, yes, it could be convergent evolution. And one point I would just raise, and this is raised a lot in the book, actually, is that these things have not been found in many of the intervening species, particularly in amphibians, suggesting that perhaps it's something that just evolved many times, that you get it in lamprey, but you get it in mammals, you get it in birds. And that's possible, but I do think it's important to acknowledge that I don't think anyone has studied sharks and amphibians with the same kinds of techniques that Grillner and his group applied to lamprey, or that people apply to zebrafish or mammals. And so I think it's necessary, before we can answer that question, to look for these things in these animals, along lineage, more studies of reptiles, etc. Because it seems to me that the prospect that it's actually a conserved bow plan, and here I don't just mean the genetic, but also the functional and the connectome aspects of it, may actually be very old. And I think that's very important for people like me who want to speculate about function. And it's because, well, if the basal ganglia was conserved since here, and its structure has been essentially laid down since this point 500 million years ago, then it's not about economic decision making or other kinds of things that we study in mammals. It was a circuit subserving the kinds of things that animals did way back here, just when they became mobile and predation took off. Anyway, so I'll stop with that. I'll stop sharing, and we can discuss. I'm hoping everybody could hear me.
Yeah.
Now that I finished, okay, good.
Clear.
All right.
So we had some questions from the audience. I don't know if Luis or Maria want to comment or add anything. Otherwise, we can invite.
Let's go to the questions of the audience. At least for me, that would be Luis?
Okay. So we will invite Daniel. Sorry, Daniel. Are you still there, Daniel? Can we invite you to make your question? He's connecting.
Yeah. Can you hear me?
Yes.
Yep.
Yeah. Yeah. My question was like, and that's the thing that I repeatedly like comes up in discussions on this topic is like, if neuroanatomical definitions should be based on connectivity or developmental lineage, right? Like, and I think that goes very well to the last presentation here. Like, and the viewpoint I'm coming from is more like, if you look at how the situation is in bioinformatics in terms of reference genomes, right? Every organism we can work with, there is a reference genome that is like the standard sequence we have agreed on we can use as a reference. But then on top of that, we have the annotation files, right? That like specify how certain genomic features are like positioned on the genome. And it seems to be, to me, the case that we need some sort of resource like that, computational resource across the evolutionary tree, but neuroanatomically. So how can we arrive at such a definition, like these kind of like computational definitions, like files, if there is like not really an agreement of how we should base the regional definition to start with, if it should be based on connectivity or developmental lineages?
Yeah, well, I've faced this question too, because I walked into this field not really knowing that much of the primary literature, and I encountered a number of debates between groups of people who really know a lot what they're talking about, and I faced myself with the question of who am I going to believe? Am I going to listen to Grillner and then not listen to Striedter and Northcutt? I mean, who the hell am I to make such a decision? So yeah, I think that's tough, and I think it's just, it's a bit of a, you just kind of hope that some convergence happens. I mean, I think regarding the columnar versus the prosomeric model, I mean, I think among the people that know the prosomeric model, even people who like to disagree with Luis on everything else will often say that it's a pretty good model. I mean, I think, I sort of feel it should start being part of the textbook so that more people are aware of it. Because again, most people I think that are aware of the debate acknowledge that it's a pretty good way of looking at it.
Right, but I also think like I would encounter a lot of people that are not aware about the debate, but are still contributing a lot in neuroanatomy, and especially I can see people that come from computer science background into and start doing a lot of like image processing and making atlases. They will start having a viewpoint where like, oh, forget about all of this like debate, and let's just like transform brain section data into like stereotactic space or something like that. So we should just talk about coordinates and do computer science as normal. And that seems to be like probably where we don't want to end up in, because that's completely like ignoring evolution and a lot of other things.
Well, I think topology is something people I think care about more than coordinates, right? I mean, people look at mouse brains versus primate brains in terms of topology. And yeah, so I think that's pretty good. It's just that you don't know, you don't always know what matches, what corresponds to what. I think that's where development actually helps, right?
Right.
See where the topology came from. And if it came from the same source along the same way, even if it looks really different, it makes some sense. A function, of course, is a whole other can of worms, but
You may speak. I mean, there's a big discussion, classical, about whether you can or how can you introduce function into considerations of homology. I think that you can, but you have to go step-like. First, prove morphological homology, developmental morphologic topologic homology, which includes genetic background, and because that is what demonstrates the problem. Actually, Once you get into that decided, then you can move into connections, and through connections you may go into circuitry and finally into function. But you cannot go directly from not knowing anything directly into function and decide that because flies fly and have wings and birds fly and have wings, then the wings of flies are homologous to the wings of birds. That is not doing things properly. You can do it if you want, and many people do that sort of reasoning, just playing with similarity, functional similarity, or some other property similarity. It can be expressing of a gene or expressing a protein or whatever. If you don't proceed in the entire way, first the topology, the developmental pattern, the genetic background, and then when you go into connections which needs connectivity of axons, axonal navigation, synaptogenesis, there are many other things, mechanisms involved, and even then you will reach common functions. And the moment when you find that the same place in the brain produces a comparable function, then you can say this function is homologous to that function. And probably in that sense, but many cortical functions, many basal ganglia functions, and many other hypothalamic and visceral control functions are homologous across enormous number of animals. But you need to do it, I think, in that particular way. You cannot jump over anatomy, over embryology, and say that this thing is irrelevant, and now I just look at the function. But then most probably you will be wrong. That's what they think. Not necessarily, because maybe you are right, but you could be wrong. That's a problem I see.
Yeah, I agree. I mean, I think the problem is many of us are just not patient enough to have all that foundational work, and we want to just start thinking about those functional issues. I'm speaking for myself because I do try to talk about functions, and I understand that there's these pitfalls. But in the work of Grillner, I think kind of hits a nail on the head. I mean, if the developmental and connections are there, and you actually go and you record from those neurons and you stimulate those neurons and you do lesions. I mean, you do the whole thing, and it works like it does in across these different species. Then I think you've got a pretty good argument. Zebrafish people, I think, do this really well too.
I think Maria wanted to add something.
No, I wanted to add to what Luis said, and I think going beyond just a comparison of progenitor domains and development is very useful to understand novelties. This is something we've not talked much about, but when we try to infer homologies, we look at similarities as a criterion to identify homologies. We also look at the phylogenetic distribution of a trait, and if a common origin is the most parsimonious explanation for similarities, then we conclude this is homology. But there's the other phase of the coin is understanding novelties, because things change. They tend to change. The more you look at, there are good examples of understanding novelties. For example, nice work on the neural crest from Marianne Bronner's lab recently, where they mapped the genes expressed in neural crest in different vertebrate species, and you see that novelties sometimes emerge from stepwise changes of genetic of gene regulatory networks and genetic interactions, and I think if we expand our view on our comparative approach to anatomy of the adult brain, cell types, and their connectivity, then we have better tools to understand differences and to understand novelties, as they, to understand how these fundamental brain regions identified by the prosomatic model have actually changed within themselves in these different vertebrate species. Because ultimately, these vertebrates are using their brain to adapt to different environments and to do different things. So there are differences that we need to understand and to explain.
You are muted, Luis. You have to unmute.
In two species, and in one of them there is a mutation and you obtain a novelty out of what was before two things which were homologous in the precursor species. And now you get here a novelty. So does this novelty cease to be homologous, or it doesn't continue to be homologous while being a novelty? Because I get the impression from the book of Striedter and Northcutt that they seem to believe that once a novelty appears, they are no longer homologous, they are now new. And newness does not come out of nothing. So if you produce a novelty, you have used something that was previously homologous to something. And I don't think that you cease to be homologous, you are just transformed. And you have new properties, you have emergent properties. But you don't cease to be homologous as you were before, from where your primordium came from. So, for instance, the cells of the pontine nuclei or the inferior olive in mammals, they come from the rhombic lip. And when you look back, what is homologous in a lamprey brain to the rhombic lip, there happens to be something expressing genes characteristic of the rhombic lip, although you don't have any migrations, typical migrations coming from the rhombic lip. So that from the point of view of the primordial rhombic lip presence and genetic position and genetic identity, you have an homology there. But now you have a novelty, which is in mammals, the inferior olive and the pontine nuclei. So are they no longer homologous with the rhombic primordium? They are derivatives from that. So that's the idea of field homology. That's where the field homology comes in, to take in the derivatives, the novelties, the variations, and all these things that come out of a primordium that was homologous continues being homologous in this novelty way, in an American new aspect. But I don't believe that you lose homology ever. So I think that all parts of all the brains are homologous, have their homologs somewhere. That may be more difficult or less difficult to find them, but I don't think that any homology can disappear, because the genome is conserving the basis of everything. So you are not losing the origin in your genome. That's the point.
Maria?
I think my take on what Luis just said is that we need to be extremely precise on what we are comparing and what kind of homologies we are inferring. So if we want to speak about the homology of a rhombic lip, which is a progenitor domain, then we say the rhombic lip is homologous. If we want to talk about the homology of the pontine nucleus, which is an adult differentiated nucleus in the brain with precise neurochemical characteristics, input-output connectivity, then we talk about that. So I think it's still possible to say that you have an embryonic field that is homologous across species, but their adult derivatives, there are some novelties, and there can be a nucleus. This is a novelty in a certain species or in a certain evolutionary lineage, and does not exist in other animals. I think this is, as long as we're precise with the definition, we can still talk about homologies and novelties, and I don't think that everything is homologous to everything, and because.
But you have 100% novel things in the brain evolution, 100% novel, just coming out of without any other antecedents. I would doubt that you can have that. Everything has an antecedent.
I didn't say it's 100% novel, but I'm saying we need to acknowledge the differences and also talk about novelties. That's what I said.
It exists, so when emergence exists, so there is a process where when a system becomes more complex, new properties emerge. So this is characteristic of the concept of a system. Whenever a system acquires more parts or more levels of organization or more subdivisions or whatever you want, increase of complexity leads to emergence of novel properties, among which you have functional properties, of course. So novelty, yes, is a result of increase of the complexity of the system. But doesn't mean that this system that is not built on the previous one, which was simpler. You just need new parts that slowly, as development becomes more complex and more possibilities are there, more options, then suddenly where one territory was divided into 3, now it gets divided into 7. And now you have 7 parts. That's how you get 50 areas in the cortex in rodents and 280 in humans, out of the same primordium of dorsal pallium. So how is that you make, out of 50, you make 280? because the territory has expanded, and once there are many more cells, then there are mechanisms that will divide the available population, cell population, in order to produce more partitions. You get a more complex cortex, and now you have novel functional properties of this more complex cortex, and then you get human mind instead of the rat mind.
I think a lot of these questions would be much easier.
So novelty, of course, is there, but this has strange ways to appear, because we don't control very well. You cannot predict where emergence is going to occur. We cannot calculate that. We just see it, recognize it when it appears, but we cannot predict that this is going to become twice as big and differentiate into this and that. We have this has to happen, and when this happens, then we say, okay, this is a novelty, interesting.
I think this would be much easier for us to tease apart had there not been so many extinctions. I think the problems always arise because we're just missing too many intermediate steps along all the lineages that we're trying to do. And so in a way you can say that, yes, of course, nothing ever comes from nothing. And there are those things, it's just that we don't know, and it's difficult to know. So in cases where we do have a very, very, very bushy tree of animals that are closely related, then we can really do this. So in primates, we do have a very bushy tree with we don't have sparse lineages that only lamprey and hagfish have survived, So it's tricky for some animals, and I wish we could, reincarnate all of them.
Let me just round off the question. Since it started, it's will it be possible to create the equivalent to reference genomes, but for atlases? It seems to be the case that here, homology is extremely important. Whereas in reference genomes, homology is the last thing you would add to the annotation, right? And usually it's also very iffy. Sometimes it's not correct and it doesn't really matter. But the basic thing is you have the reference sequence of the genome, and that usually is fairly trivial. In the case of reference atlases, it's very trivial for anatomy. It's just space. But on top of that is the annotation, right? Where gene bodies are, and where do you have the 3' UTR of this gene and stuff like that. And it seems to be the case here that we are not clear on that terminology at all. What are the essential features of neuroanatomy? Because if you would be clear on that part, then homology can just be added on top as we're learning more, but we are still agreeing on that these are the essential features.
Idea of a Bauplan. The Bauplan concept contains whatever seems to be essential because it happens not to change over millions of years. Why does it not change? Because every time it tried to change, the survival went down. So that's why is it so continuous that you have a Bauplan, because you need this Bauplan in order to stay alive in the first place. And once you have this Bauplan there, you can be alive, and now you can try to hear better, or to see better, or to move better, or to do this other thing better. Or you can introduce a completely new function. I hear that, I'm not a specialist, that in butterfly wings, the initial function of the primordial the wings of the butterflies was to refrigerate the blood of the animal. And that's why they expanded in size and become flatter and increased surface, because in that way they become better refrigerators of the blood. But suddenly they found out over a given threshold of surface, suddenly they got elevation and they could fly. So that's the emergence of a new function, completely unexpected, because the animal was just trying to refrigerate better its blood. And suddenly he obtained flight. So that tells you in which sense function relates to morphology. It's difficult to predict and difficult to say that there was a plan for something. You just advance in a given direction of evolution, and suddenly a new property jumps up. And suddenly you have a novelty there. It can be derived from what you had before, or it can be a completely new thing, like flying in butterflies.
I think this is a theme we're going to come back to in further chapters, because the major transitions, like getting out on land or becoming nocturnal or becoming warm-blooded, these things were associated with major neural changes that are very specific. Why we have a cerebral cortex has a lot to do with the fact that we hid from diurnal dinosaurs for 200 million years or so, and just became took a different strategy. We became these little nocturnal things that scurried around, and then from that baseline, we had a different path that we took, very different than what birds have done. And I think again that's why this book and this approach I think is so valuable, because otherwise it just doesn't make sense. Why don't we do it the way the birds do it, or other animals do it? And it's the same story for them, of course, right? So again, I think that that's going to be the theme that's going to be the question for all the chapters.
Okay, so we are 4 minutes over the 2 hours we set. We have other questions. I don't know what should we do.
They seem to have disappeared.
The questions?
I don't see the questions anymore.
I don't see the questions.
Oh no, there they are. Okay, sorry.
Yeah. Sorry. I don't know if you guys have time for the last question.
I can stay for a bit, sure.
Yeah? Okay. Thank you, Daniel, by the way, for your question and your comments. So we will ask, we had the question by Joe Graham. He cannot come up on stage. I don't know if you want me to read it or you can read it yourself. Maybe that would be faster. Can you see it?
Maybe you can read it, Manuel,
okay,
for the
The CNS is a hollow, closed tube that seems to me to have evolved the same way it develops in the individual animals, starting as a hole in the head and eventually extending to dump into the gut. Neurenteric canal, before closing off both ends. Can you discuss how the CNS evolved structurally and what evolutionary advantages the developing tube structure would provide? Any comment?
I know what is people call the neurenteric canal, and I've looked at several pictures and drawings of it, and I never could make much of it. I can say that you move in the caudal-most area of the body, where you are not sure in the embryo, you are not sure whether you still have neuroectoderm or not, where you have only skin, where you have endodermal. So all this territory is uncertain from the point of view of interpreting which cell types you are dealing with. So calling it neuroectoderm is just a name, but it's just a caudal transient structure that seems to connect the cavity of the neural tube caudally with the cavity of the gut, of the caudal gut. That's why it's called neurenteric. But I don't confide that this is really significant. I think that's just part ignorance of the observers, that they don't know what they are looking at. That's my impression. And nobody has seemed to have studied recently this area of the embryo. Nobody looks at the caudal end of the embryo. Everybody looks at the cortex nowadays.
Well, regarding what evolutionary advantage, I think that's a good question. Actually, I've thought, I've wondered about that too. We had this neural plate, which seemed to be a pretty well organized system. At some point, well, first of all, there's the question of how it came to be dorsal rather than ventral, which is a pretty crazy story too, with lots of debates. But why did it fold inside? I mean, was it for protection? I don't know if anybody's really has a good. I myself haven't read. I was hoping maybe Maria or Luis would be able to say something about that.
Not much, honestly. I don't know if I think there's probably some causal relationship between the folding of a neural tube and the dorsoventral inversion, because the position of the mouse in chordates it is not on the way, let's say, of the neural tube, and instead, if you look at the invertebrate nervous system, you have this. Take, for example, the classic nomenclature. You have the supraesophageal ganglion, and then these nerves that wrap around the gut. So the gut kind of comes in the way, let's say, of folding a tube. because it's nested in between. Because the mouth opening is the derivative of the blastopore, and once you have dorsoventral inversion, there is essentially the blastopore gives rise to the anus and not to the mouth anymore. You have a new position of the mouth, and then all the neuroepithelium is not interrupted anymore by mouth, and I think that might have been permissive for folding it in a tube, but the reason why. The advantage of having it in a tube is also not very clear to me.
So you mean it's hard to understand why it folded in the dorsal neural plate, but we still don't know. But at least we can say that in the case of a ventral one, it would have been difficult. Okay, I think Luis wants to say something.
I wanted just to comment that you cannot ask why evolution occurs. I mean, evolution, that will be teleology. So evolution does not occur for reasons. There are no reasons. Simply things happen, and it's not one or two things, just the generation before. It's thousands of, hundreds of thousands of generations before. What happened during all these small minimal changes to morphogenesis and to developmental patterning, accumulation of millions of small changes here and there in the genome, that is what leads after millions of years to evolutionary change.
Of course.
So it's false to think that just because this fish needed to survive, then it moved into Earth because it had a very strong predator situation, and he had to fly from the sea and move into Earth, and he developed the legs. That doesn't happen that way, because the legs appeared during many million years, and the animal that was developing it didn't want to go out of the water. It just developed the legs because it had to develop the eggs.
Then we have a huge selection bias. We can't
function for them, but you cannot do teleology. If I criticize the book, if I can make, it's just in that sense that in some chapters or in some conclusions. Not that they also defend teleology, because I know them personally, I know they don't, but the way the text finishes some idea, some reader might get the idea that it's being defended that this novelty appeared because this animal had this problem and needed this solution. This idea came when I read the book, and I read it thoroughly. I got this, and when I commented on the book to the authors before it was published, I commented on this topic. So I told them, I think that here it reads as if there is a problem in this poor animal, he had to find the solution, and eventually he found it. And that's not the way of evolution. I'm sorry about that. But, so I don't think that the authors, I'm sure they are clear about that themselves. But so that they absolutely come with me in this point. Absolutely.
I think it has a lot to do with the way we speak. We always speak with words that convey purpose, even if that's not what we mean. I almost feel like they should have a footnote on every page that just says, there's a little star, and then on the bottom it says, by the way, we're not talking about teleology. And then just put that star in half the sentences, because we know that they don't think that, right? But once they start saying, it's like they just need a big caveat in the beginning saying. That's a hard time.
Okay, we are 10 minutes over the time we said in the beginning. Maybe we should stop here. I think it has been a great discussion. I'm sorry if we didn't answer all the questions, but there were many comments, interesting comments, and I hope you all have a great time as we did. Just say goodbye to everyone. Thank to everyone. We will be back on the 2nd of February with the 3rd chapter, in which I think we discussed vertebrates. I don't know if Raquel or Alex want to add anything, but if not, we will.
Well, just thank you all the speakers for your assistance, and all the public also the assistance for being here and participating.
It was a very interesting session. Thank you.
Okay. Bye, everybody.
Thank you.
Thank you.
Thank you, Maria. Thank you, Paul. Luis.
Thank you, everyone.